Hello everyone, so continuing with plant reproduction video part 2. We were going over the parts of a flower, which is the reproductive organ of a plant. Flower is a modified shoot. It is the modified stem, the tip of the stem of the plant, and the parts of the flower are modified leaves. So this is an adaptation for reproduction. We looked at the 4 rings, concentric rings of the flower, going from outermost to innermost. Outermost are sepals, and then we have petals. They have different functions. Then is the male part known as the stamen, and then the female parts in the innermost area are the carpels. We'll identify some flower structures. So here are the sepals. This is the petal. We are looking at the anther and the filament, which is a part of the stamen. I can count 6 stamen here. This is a lily flower. Over here also I see the 6 stamen. And then here is the stigma, style, and ovary would be deep within. Here we see the stigma, style, and the ovary will be deep within. So these are lily plants, lily flowers. Here is a sunflower or a daisy. There are many variations of flowers in angiosperm. We see this one here is a sunflower, which is not really one flower, but it is a collection of tiny florets. And the sunflower is really an inflorescence. It's a collection of flowers. Here we see a collection of flowers, just as we see here. Here is an orchid, and here's a daffodil. Now flowers come and they show different kinds of symmetry. Here is one with a radial symmetry, which means it has multiple axes of symmetry. And this one here, orchid, has bilateral symmetry. The flowers with bilateral symmetry are more advanced or more successful as compared to flowers with radial symmetry. And we will go over that in our next slides. Here we are seeing, looking at the flower for flower of a corn. The male and the female flowers are separate. They are located on the same plant, but the female flowers at the base of the plant and the male flowers are at the tip. The flowers don't have sepals or petals. They are just comprised of carpels if it's a female flower, and then or male flowers would just have stamen. We are looking at the stamen, and they will produce pollen. And the female flower, all the carpels will be enclosed in one inflorescence, and these are all the styles. And the stigma, the silk of the corn, is basically stigma and style of multiple carpels. And once they are fertilized, then you get the corn on the cob. Each corn kernel is a seed, which is a product of fertilized ovule. So flowers and pollinators, they co-evolved, and here again we see a flower with radial symmetry, and here is how it would be visible to a bee under UV light. So bees can see UV light, and they can see how the center of the flower is the bullseye to the nectar, and that's what is going to guide the bee to the center of the flower to get nectar. And while it is getting nectar, it will also pollinate, cross-pollinate the flowers. So radial symmetry would be an example for this kind of flower, and then this one here we see is bilateral symmetry. Because most pollinators have bilateral symmetry, for example, this hummingbird would have bilateral symmetry and the bee would have bilateral symmetry. So the flowers that have bilateral symmetry can be specialized to have a specialized pollinator with the right body shape and size in order to optimize pollination in exchange of nectar that the pollinators do come to the flowers to get. So coevolution of these animals along with the evolution of plants have influenced each other. For example, animal herbivory, when animals feed on plants, that has naturally selected plants with those specific defenses. Defense mechanisms would include having thorns or producing sticky exudes or having tissues that are offensive, or like a chili pepper, it has capsaicin, that is the spicy taste, so other animals, herbivores, would avoid them. So those are plant defenses those are going to be naturally selected against animal herbivory. So herbivores and plants, they co-evolved and they have those traits that are influencing each other. Similarly, pollinators and flowering plants, they have co-evolved because they have mutualistic behavior, they benefit from each other, they exhibit mutualism. Flowers with bilateral symmetry, they restrict the movement of pollinators and therefore they will target and maximize pollination, and hence would lead to speciation of a specific variety of a flower or plant, and speciation of the pollinator as well. In this slide, we are going to look at the development of the gametophytes, both for male and female gametophytes inside a flower. So the tissues of the flower are diploid and they are sporophytic tissue. We will look at the development of a gametophyte within a flower. Please know that the entire plant is a sporophyte and is a diploid. Within the pollen sac, the fancier name for the pollen sac being microsporangium, micro meaning small, sporangium meaning a container that contains spore. So microsporangium is a fancier name for the anther lobe or the pollen sac. Inside the pollen sac, there will be microsporocytes. Again, micro meaning small, sporocyte meaning a cell that will produce spores. So microsporocyte, this is a diploid cell. It's still a sporocyte or part of the sporophyte. That undergoes meiosis. The sporocyte, the diploid sporocyte, microsporocyte, undergoes meiosis to form 4 microspores which are haploid. Next we see each of those 4 microspore, it develops into a pollen grain. This pollen grain will divide once by mitosis to form 2 cells. One cell forms the generative cell and one cell forms the tube cell. The generative cell nucleus divides one more time by mitosis to form 2 generative cell nuclei. The generative cells will be within the tube cell, and the tube cell will continue to grow when it lands on the stigma of the flower, and we will see it in our next slide where the generative cell nuclei will travel along with the tube cell. So that is the development of a male gametophyte in the pollen grain. So at the end we have a male gametophyte which has 2 cells and a total of 3 nuclei. One nucleus is the tube cell nucleus, and the generative cell will have 2 nuclei. So 2 cells, 3 nuclei. That's why it is a multicellular male gametophyte. Every single nucleus is going to be haploid. Let's look at the development of the female gametophyte, also known as the embryo sac. The female gametophyte will be inside the megasporangium. Mega meaning large. Sporangium means a container that contains the spore. So megasporangium, a fancier name for the ovule, the entire ovule. In the ovule, there will be the megasporocyte. This megasporocyte is also diploid, and this is the cell that will undergo meiosis. So it's like the mother cell that will undergo meiosis. We see 2 integuments, and we see an opening over here, a pore in the integuments, which is the micropyle. Micro meaning small, pyle meaning an opening. So the megasporocyte undergoes meiosis, and there will be 4 haploid daughter cells. Out of the 4 haploid daughter cells, 1 will be rather large, and the other 3 being rather small, they will perish. The single surviving megaspore will divide 3 times by mitosis. There is a difference at the end of meiosis in megaspore production versus a microspore production. When microspores are produced, there will be 4 equal sized microspores, and each one of them will develop into a pollen grain. Here, out of the 4 megaspores, 1 will be rather large, will have more organelles and nutrients. That will be the one that survive, and the rest 3 will degenerate. So unequal meiosis in female gametophyte versus male gametophyte is the same as in animals too. Sperm production, at the end you get 4 sperm from each spermatocyte, and for each oocyte there is only 1 surviving egg. The rest degenerate or perish or become polar bodies. Let's continue on and see the survival of the megaspore that remains. It undergoes 3 mitotic divisions. The surviving megaspore undergoes 3 mitotic divisions. So from 1 cell it becomes 2 cells, and from 2 it becomes 4, and from 4 it becomes 8. So we end up with 8 nuclei inside this thing known as the embryo sac, also the female gametophyte, and then cytokinesis will divide it into individual cells. We end up with 7 cells because the central cell has 2 polar nuclei. The nuclei from the 2 opposite poles, they migrate to the center and become a single cell, and at the opposite ends there are 3 antipodal cells, and at this end there are 3 cells, the 2 synergids and the central egg cell. So that is the female gametophyte, 7 cells, 8 nuclei. The male gametophyte has 2 cells, 3 nuclei. So multicellular and haploid. And that is the development of the male and the female gametophyte. In my next lecture, we will look into double fertilization. Thanks for watching.